So welcome back friends, welcome back in another video tutorial of protein engineering. So today we are going to discuss about various types of site directed mutagenesis. We have already uploaded a detailed video on site directed mutagenesis. If you have not any kind of basic knowledge of site directed mutagenesis, then I would like to recommend you to go and first watch this video and then come back here. Then it will be helpful to you to understand what is going on here. Okay, because here I will not going to touch the basics of the site directed mutagenesis. I will only teach you here that what kind of modification we can done with the help of site directed mutagenesis. Okay, so let's get started. So here we are using a plasmid for all kind of manipulation we are doing, although you can also done such kind of manipulation with help of gene of interest also. Okay, so the black circle here is indicating the double stranded plasmid. Okay, and these dotted lines are the thing that will get after the mutagenesis process. Okay, so the first thing that we can do with the help of site directed mutagenesis, and we have also discussed about the first example of site directed mutagenesis, that is the point mutation, in our details video discussion, because here in that video we have directed a point mutation in a protein, right? I hope you can remember. So let's recapitulate what we have done there. So another important thing I want to tell you, that all kind of site directed mutagenesis are also known as PCR directed mutagenesis. Okay, because here we are simply using PCR to perform those kind of mutagenesis. Okay, so now let's start. So as I think you know about that, PCR requires two kind of primer, forward primer and backward primer, or forward primer or also known as reverse primer, right? Two kind of primer, forward and reverse. That is what reverse. Okay, so in case of point mutation, the idea will be the reverse primer will be exactly on the basis of its complementary DNA template, but the forward primer will be, we will construct the forward primer in such a way that, suppose if the forward primer is of 25 base pair length, then 24 base pair or 24 nucleotide will be exactly complementary of its template. Okay, but one nucleotide will be of our choice. Okay, that exactly we have done in case of when we have discussed about the site directed mutagenesis detailedly. So this one misincorporation of nucleotide will facilitate the formation of a DNA of interest, which will ultimately transcribe and translate to produce a protein that will have a mutation at that particular position. Okay, depending on at what position we have incorporated the mutation. Okay, for example, by changing only one nucleotide, we can insert aspartic acid in place of, for example, let's say leucine. Okay, with the help of that point mutation. So again we are here incorporating a one nucleotide which is a mismatched nucleotide that will facilitate the production of a protein that I should say it is a recombinant protein, which will contain a amino acid of our choice in place of the normal amino acid. Okay, so that is the first kind of site directed mutagenesis that help us to undergo a point mutation. Okay, now let's move to the next slide. So that is the deletion. We have not talked about deletion yet in previous video lectures, right? So here the idea is very simple, because let's try to focus in the picture. If you will focus in the picture, the forward primer and the reverse primer are incorporated at the end, right? That means for where forward primer is ended, At that particular position we have incorporated the reverse primer, right? That means they are attached end to end. But in case of deletion mutation, the incorporation of forward primer and reverse primer is not end to end, because there is a small gap in between them. Okay, so as a result, when we will run the PCR, the DNA template, that is the part, this part of the DNA template will be remain unread by the DNA polymerase, because here we have not provided the primer, right? And as a result there will be a deletion. So that is how, with the help of site directed mutagenesis, we can incorporate deletion. One thing, please remember this very carefully, that in case of point mutation you have to attach the forward primer and the reverse primer end to end. But in case of deletion, when you are willing to incorporate a deletion, there should be some gap between the forward primer and the reverse primer. Okay, so in point mutation incorporation, you have to mutate your primer at a single base. Okay, so now we are going to discuss about how we can insert a short stretch of nucleotide with the help of that site directed mutagenesis. So here we will add a primer, that is forward primer, in such a way that it will contain a short stretch of nucleotide extra. Okay, that means up to that part it is exactly complementary to our template. That is why it is wellly annealed, but it remain as unannealed, right? So suppose if we are using 25 nucleotide primer, so we have added extra 5 nucleotide at its end, that means a primer of 30 nucleotide, but our DNA template will recognize only the 25 nucleotide. So as a result, the 5 nucleotide will remain unannealed. Okay, and we will also incorporate the reverse primer. Then after replication during the PCR run, it will be the unannealed part will also be copied by the DNA polymerase. Here we are using Taq polymerase. Okay, so Taq polymerase will also copy that unannealed extra part of the primer. Okay, and as a result, after the ending of the PCR, we will get a product that will have that extra part of the gene as a replicated part. Okay, so that is how we can insert a particular stretch of nucleotide with the help of site directed mutagenesis. But remember, it is only the option number 1. That means here we are incorporating the short stretch of nucleotide that we want to insert at the end of the primer. Now I am going to discuss with you another possible option, where we will going to construct the primer in such a way that the extra part will be at the middle. Okay, so let's see. So that is the idea. Okay, so here we are constructing the primer in such a way that the extra part will be at the middle. Okay, so as a result, they will form a loop like structure, because they will not get any kind of complementarity with the template, and as a result they will convert a loop like structure. And we have also incorporated the reverse primer. Then after PCR we will get that extra part of the DNA as a replicated product. Okay, so this was the insertion option 2. So that is the various kind of site directed mutagenesis, various kind of thing that we can do with the help of site directed mutagenesis. We can incorporate a particular stretch of nucleotide in any kind of position that we want, right? By incorporating that particular nucleotide either at the end of the primer or at the middle of the primer. Okay, and we can also do a deletion by attaching the forward primer and reverse primer by keeping a gap in between them. Okay, and the first very one that we have also discussed detailedly, that is the point mutation. We can incorporate this with the help of site directed mutagenesis. Okay, so this short video is just about what we can do with the help of site directed mutagenesis. That means here we have only discussed about the application of the site directed mutagenesis. So I hope this video will be helpful to you, and thank you for listening to this class.